name is Mara Carney. I'm in Dr. Melissa Bailey's lab, and what I did last summer was I induced a bunch of female mice with diabetes using streptozocin. What it does is it attacks the pancreatic cells in these mice, and it induces diabetes. What we did was we mated these mice with male mice of the same type, and we were looking at the ability of these female mice to be able to keep their pregnancy, and we were looking at differences between the maternal weight and the fetal weight of our experimental group and our control group. Our control group was not diabetic. So what we found was there was really no difference. We had a very, very high blood glucose level with our diabetic mice. However, our non-diabetic mice, our control group, they had pretty normal fasting blood glucose levels. The results with our fecundity, or the ability to keep their pregnancy and the ability to maintain the pregnancy, there was really no difference. So it's important in our lab because we do a lot of diabetic studies, and we use streptozocin quite often. We also do a lot of developmental studies. So we were trying to see is if it was a viable option for us to use in our lab for future studies.
Okay. Were the results surprising?
The results were kind of surprising. We did not use insulin in any of our study to maintain blood glucose levels. We wanted those high blood glucose levels with our female mice. So we didn't use insulin. Literature shows that we should use insulin to maintain good blood sugars in order to maintain pregnancies. However, our not using insulin, our high blood glucose levels, they really didn't show much of a difference in the ability to maintain a pregnancy. So that was surprising to us. Also, typically with like human patients with diabetes and females that have pregnancies, usually they have huge babies, 10, 11, 12-pound babies. We didn't see a difference in the fetal weights of any of our mice. So that was also kind of surprising to us.
What we wanted to do with our experiment was explore the impact that context has on the way you remember and the types of details that you remember. So what we did is we had 107 participants, which we divided up into 3 equal groups or as close as possible. And the first group that we had was we had individuals view 12 different objects one at a time. And for the second group, we had individuals view objects in a small group, so 4 different objects. And then for our third group, we had participants view the objects simultaneously, so all 12 were in the context of a scene. And then what we did was we asked some questions which pertain to the type of details about the objects and the things that they would remember. So, for example, some of our measures were quantity. Another was the words on the products or the objects that we showed. Another was specific characteristics for these objects. And the fourth measure was color. So what we did is we had a hypothesis that the context which you view an object in will affect your total recall score based on those 4 subscores. And we found that with the one-way ANOVA, there is a significant impact on the types of details that you remember based on the condition which you were placed in. So for example, individuals in the small group had a percentage score of about 85, but then if you go and look at those in the scenic group, they had a percentage of about 45, which is a significant difference. I mean, you can think it's about double the score. So we were like, well, you know, what accounts for this? Is it the way that you have to space your attention out when you're viewing one object versus 4 versus 12? Is it the fact that maybe you don't remember things as accurately as you thought you did when you view them singly, and you have to pay attention to here's the first object, here's the second object, and what comes after that? But that's what we did, and we also ran some correlations, which my friend analyzed, and he found that details. Because remember, there were no negative relationships between subscores. So I think that's interesting because there was no certain type of detail encoded which negatively impacted an individual's ability to remember other types of details. So ultimately, we think if we were to do this in the future, that what we could do is find some sort of stimuli which is neutral or completely different, because what we did is we used our schema for this was office supplies. It's something that students would encounter on a daily basis. It's something they're familiar with. And we are willing to acknowledge the fact that that may have frightened them because they came into contact with objects that we used, which could have had a positive or a negative impact on our study, depending on how you look at it. That's what we did.
So the results were expected, surprising, what?
Well, the results were a little bit surprising. We were definitely looking for a number one. However, we thought that was going to be an individual object because you're only looking at one object and you're staring at it, you're going through and you're looking at only a single object. However, it wasn't the single object that won out. It was a small group that took it by storm. And I'm really curious to figure out why people will remember more specific details about 4 objects rather than just thinking about one. And currently, I was really grasping for some sort of discussion topic on what might have accounted for this. And I did think of a few ways about how we did our scoring methods. Had we gone through and asked them the questions about the individual object directly after they've seen the pictures, instead of after they've seen all 12 slides, that may have significantly impacted the amount correct they have. However, we couldn't do that because you can't normalize that across all of our groups, because you have one for the 12 objects, for the scenic objects. Well, you can't split that up 12 times. I mean, you just have to sit there and look at it once. Or for the group objects, you can do that after they've seen each group. But like I said, 12 times versus taking different things 3 times versus taking it once. If we could find a way to redesign the study to normalize that, I still have some faith that the individual object group would probably take the highest total amount correct of recall. If we could figure out, if we could design and redesign the study to call that into question.
So it's just an application that's happening now.
So we're talking about eating disorders and art therapy. And these are some directives that are done at a treatment facility for eating disorders. At the treatment facility, they integrate individual, group, and art therapy. So this one is called the Separating from Eating Disorders, and the clients are asked to draw their eating disorder separate from themselves. So what it would look like if it was its own person. And so this is an example. And then they're also asked to draw their true self without the eating disorder. And then they're asked to draw an environment to have the eating disorder and their true self in. And this is kind of a way to show their progression during treatment. And so right now this person is just starting treatment, and so their eating disorder is still taking over their life and their true self. But the goal is for the true self to move away from the eating disorder, and then the eating disorder to just be completely out of the environment so that the true self reigns over the environment. And it's just a good way to show the clients that they are not their eating disorders, that's not their identity, and it helps them separate the eating disorder from themselves so that they can recover. Did you want to talk about that?
Sure. It's all, as well as doing it individually, you can also do it as a group directive. One of the ones that we chose to focus on is called the mirroring activity. It's a way to have the clients feel what it's like to not be in control, because a lot of with what's going on with eating disorders is that clients feel like they are constantly needing to control what's going on with them in terms of monitoring their diet, what they're eating, that sort of thing. So the way that this activity works is you'll have 2 clients sitting across from each other. One will play the role of the leader and will start drawing. The other will play the role of the follower and try and mirror what they're doing. And then they'll switch roles. and then process what it was like to be in the role of the one who's in control and the role of the one who's not in control, and then just see how that, you know, feels with the treatment.
Well, maybe if you're related to maybe you're related to that. So they help people with geological research.
I'm going to start off with a quote. Designer of the Spaceship One, Burt Rutan, said that testing leads to failures, but failures leads to understanding. And this is never so true when it comes to research. You have so many failures and very few successes. Today, thankfully, I have something to share with you, and it's a success. To go about and approach and explain this, I'm going to give the, the why, the what, and the where. The why is, to date, little information is known on the regulatory mechanism involved in my protein of interest. Some information is known, however, by exploring this area, we'll get a better understanding of a eukaryotic cell. Furthermore, it'll play a better role in understanding carcinogenesis. Since cancer is caused by uncontrollable cell proliferation caused by a variant cell's lack of control, my protein of interest, which is CC14 phosphatase, could be one of the so-called out-of-control proteins and or activators that can be targeted for anti-cancer drug therapeutics. So the what is my protein, CC14 phosphatase. It's a phosphatase which removes phosphates from organic compounds. The where, it is seen highly and expressed at high levels during the mitotic cycle, specifically in the fetus. When it comes to everything else, I've gone through my various assays and purification and techniques, and I got a result between an SA's page gel, and my protein is just around 62 kilodaltons, and I got a huge band, Whopper-sized band in the 60-70 region. So I did a phosphatase assay, which tests for the activity of my protein, and it's a qualitative test, so the switch will turn clear to yellow. It didn't turn yellow, so it was inactive, and I wanted to know exactly what if I isolated my protein of interest. So I sent it off for mass spec, and they confirmed that I successfully isolated and purified it. However, it was inactive. So the future of this project is to, when it comes to purification technique aspects, we previously had an N-terminal histag. I rearranged the tag to the C-terminus, and hopefully this will change the activity of the protein. That's it. That's it.
Why exactly is this protein?
Since it's expressed high levels in it's right before the division of the cytoplasm, that is an area to where you can, since cancer is that uncontrollable cell proliferation, so if you can stop one of the steps right before the division of the cytoplasm, you could potentially stop cancer. So that's why we're interested in that specific field.
It was Chris Siegel.
Summer, thanks to USERP, we did a research grant called Zombie Survival Fitness, and we wanted to take a look at movement-based education, where we work to have students moving throughout the classroom during literacy. It was really exciting. We created a camp for 4th through end of 6th graders, and it was a week-long camp where they learned how to keep themselves awake during lessons and interact. What else do we do? We taught them strategies.
Yes. We talked about nutrition and what the brain needs to function and learn.
We published our work. We have it on our website to work on it. It's out there for teachers to use. It's research-based. All teachers know that they should be doing these things. And now it's out there for them to use it. Just click right on it and work on it.
poster is about luteolin as an antioxidant and cyclophosphamide, which is an anti-cancer drug that causes free radicals in the cells. So free radicals are what antioxidants are supposed to bind, and free radicals cause all sorts of harmful things, DNA damage mostly. That's going to cause mutations. So we've proven that cyclophosphamide, which causes these harmful free radicals, its effects can be curbed with other known antioxidants like green tea. So we want to know about luteolin, which doesn't have a lot of literature. So we go in and we test whether or not we can curb the effects of this cyclophosphamide, these free radicals, with luteolin. And it turns out that it did the reverse of what we thought it would. We thought it would make it better. We thought it would help out these cells, but it didn't. In each case, the luteolin, so our antioxidant, plus our oxidative stress-causing drug, made it actually worse. We don't know exactly why. We think it's a specific mechanism or interaction between our drug and the luteolin. Luteolin can be found in like orange peels, green bell peppers, things like that. And it's not a huge cause for concern because most pregnant women are not taking these anti-cancer drugs, but we have to use something to induce this oxidative stress. So that's why we use cyclophosphamide. We also thought we saw premature eye opening caused solely by the antioxidant. We found this wasn't true once we did further investigations. It turned out that our criteria for classifying what a premature eye opening was slightly off. It's hard because these fetuses, they're only about the size of a quarter, and it's hard to tell because mice's eyes open after they're born. So when we pull out these fetuses, it's hard to tell because their eyes are going to open for another 11 days. So what we did is we checked these fetuses over time, and we allowed them to go to term, we allowed them to birth, and I monitored their eye opening specifically, and we found that it wasn't causing premature eye opening, but it was exasperating the effects of the cyclophosphamide. Overall, this study shows us that we didn't get what we expected, but it's still valuable information for us to work off of in the future, and that we're not actually seeing premature eye opening, so we have to be a little bit more careful with our criteria.
My name is Morgan Riggs. I'm a political science student here at ESU, and my research project is on poverty, and it's something I'm very passionate about. It was very difficult doing this project, and I enjoyed doing it here at ESU. It was fun. Basically, it was hard identifying a solution to poverty because obviously there's not an end to poverty right now. I put a large emphasis on education, and education is really important. One cause for people that are falling behind in poverty is that they usually, single mothers, they drop out of college because they get pregnant and they gotta like find a job and whatnot. Another people that are like that aren't represented within poverty are children that are aged 15 and below and elderly because they don't have jobs, so they don't have an income.
Well, I did my presentation over bullying. I applied Hershey's social bond theory and Gottfried and Hershey's general theory of crime to bullying in high schools. I decided to put out a survey for all 3A high schools in Kansas. The reason why I picked high schools is because I feel like that's the age where kids should know right from wrong, if they're bullying, if they're not bullying. In my theory, in Travis Hershey's theory, he uses attachment, commitment, belief, and involvement to prove that if they have those 4 elements, they're unlikely to do crime or commit deviant acts. And in Gottfried's general theory of crime, he says that if kids have higher self-control, then they're less likely to commit deviant acts. So I said the thing, while keeping those theories in mind, I decided to make policies where The first policy will have kids being more involved in after-school programs. So I feel like if kids are more after-school programs, they're less likely to commit deviant acts like bullying or anything else. And that also gives them something productive to do with their time, besides spend their time trying to pick on kids or do drugs or join gangs. I feel like those programs should be run by teachers instead of like people they get from the streets or other teenagers. Just because I feel like if you have somebody that you can let down in your life, if your parents aren't that people, then that just gives you more of an incentive to do good and be a good person. My second theory will have teachers go to workshops every once or twice a semester, because for what I found in some of my research, that a lot of students feel like the teachers don't do enough to help them, and they feel like the teachers don't notice bullying, or when they see bullying, they don't think it is what it is. So I feel like that if teachers go to more bullying and get the help they need to see that what bullying is, how to stop it, or what they need to do to avoid it, then that will improve bullying statistics in Kansas.